Hi, Dr. Wang here. Today I'm going to help you get ready for this lab called Plasmid Mini Prep. The instruction for this lab can be found on the Commons page. So if you go to Book Lab, go to this page, Week 3 Plasmid Mini Prep, you will see lots of resources and protocol. And there are a couple of assignments associated with this lab. First of all, we need to understand the theoretical basis behind this lab. So the assignments associated with this objective is MSH2 Project 1, Pre-Lab 2. So and also a bioinformatic assignment called APE2 annotation. These three assignments will help you understand why we need to isolate plasmid DNA from the 3 E. coli strains, and what are the following steps after we have achieved today's goal. And then the last assignment associated with today's lab is called Flowchart Lab 2. This is going to help you actually perform this experiment in the lab. Even though we're doing this virtually, it's still important to understand all the principles behind the technique. So I'm going to click on this assignment. By the way, all the instructions for those three assignments are embedded in the assignment itself. So I'm not going to go over them in this training video. All right, so coming back to Flowchart Lab 2. So I'm going to do a couple of questions here just to give you an example on how to approach this type of assignment. All right, so here, the goal here is again to run the protocol right here into a flowchart. This is what we normally do in the lab. When you are handed the protocol, the first thing is not to rush to the bench and then just do this protocol step by step. It's actually what we normally do is to read the protocol a couple of times, render this into a flowchart, go through this in my mind a couple of times, and then I'm going to do this. Right? So this is going to make sure that you have everything ready when you're actually at the bench and ready to carry out the experiment. So the first step, right, so is to start 2 milliliter of E. coli overnight culture. So we can see that this is already represented on the flowchart. And then the second step is to perform centrifugation. This is going to separate the cells away from the culturing media. So here's step number 2. We're going to discard the supernatant. And then to keep the pellet, right? So you can see right here. So we're going to discard the supernatant. Therefore, fraction H stands for supernatant. This is something we want to discard. And then G stands for pellet. So this is the answer for question number 2, right? So where is the plasmid DNA? We always want to keep the fraction that contains the plasmid DNA. At this step, the plasmid DNA is still contained within the cell. So fraction G is the fraction we want to keep. So this is the pellet. All right. So, and then I'm going to continue to read the protocol and then to match it onto components on the flowchart. Right? So following this step, we are going to add TE buffer to resuspend the cell. So buffer A would be TE buffer. And then after this step, we're going to move on to this step-by-step protocol from Zymo Research. This is a commercial kit, and the protocol is provided by the company. So here, this is the protocol, right? So we start right here. We have 600 microliter bacterial culture, right? So, and then to that, we are going to add 7x lysis buffer. Right, so here we can see the step are represented right here. So we have this E. coli suspension, and then to that resuspension, we're going to add the 7x lysis buffer. So this step is going to break the E. coli cells and then release the plasmid DNA. So after this step, the plasmid DNA is now in solution, it's soluble, right? So, and then the question for you would be, right, so after the treatment, we're going to do another step of centrifugation. This is going to separate the entire mixture to fractions of the pellet and the supernatant. So where is the DNA now, right? So here, this is basically how to approach this question. There are a number of buffers used throughout the entire procedure. So here, your task is to match the name of the buffer with their location on the flowchart. Right. So, and then question number 2 is to trace where the DNA is. Is it in the pellet? Is it in the supernatant? Is it on the column, or is it in the flow through? Right. So this is what I want to do, sort of enough hint to get you started on this assignment called flowchart.